Hello everyone, myself is Dr. Satish Maurya and today we are going to talk about shoot development. In this module, we will see how the above ground shoot develops to give rise to a distinct architecture of plants with the main stem and branches. Leaves, which are also part of the shoot, will be discussed in the upcoming modules. So before moving further, I would like to discuss the objectives of this module. So the whole module is divided into three main objectives. So the first objective is the development of main shoot. So where we are going to discuss about the development of main shoot. And then next we are going to talk about the apical dominance and branching pattern. So how the apical dominance takes place or the SAM dominance takes place and the branching pattern happens in the plants. And further, or the last objective of the module will be vascular development and secondary growth. So how the xylem and phloem grows with respect to the development of a plant or a tree. So coming to the first objective of this whole module or this module is the development of the main shoot, where during embryogenesis the apical basal axis is established and this is maintained throughout the plant's life. The whole above ground body of plant is derived from this SAM through the iterative production of a phytomers. So what are the phytomers? So the phytomers are typically consists of a stem segment with an associated leaf. So this is the phytomer which is highlighted here with dotted line, where which consists of a node with leaf and axillary buds as well as the internode of a stem. As seen in the earlier modules, the primary shoot apical meristem has a central zone of quiescent cell, which is highlighted here, which is surrounded by the rapidly dividing stem cells in the peripheral zone. So that is indicated by the PZ abbreviation, and the rib zone, which is RZ, which is present at the lower side. So the peripheral and rib zone cells give rise to leaf primordia, which is highlighted here, and the future stem respectively. While looking at the molecular level, the two homeobox transcription factors, that is shoot meristem less, that is known as a STM, and Wuschel, that is WUS, which is highlighted here with the red boxes, have complementary functions in the SAM, as we have seen in the earlier module on embryogenesis. STM maintains the meristem cell fate and WUS, that is Wuschel, specifies a subset of cells at the shoot apex as stem cells. STM is expressed throughout the SAM but not in the lateral organ, that is leaf primordia, and is vital for maintenance of meristematic activity. After transcription factor asymmetric levels, that is AS1 and AS2, which is highlighted here, control the induction and growth of leaf primordia and repress STM expression. Hence, STM and AS plays a role in distinguishing stem cell and organ primordia cells. Shoot elongation in rosette dicots and monocots takes place during the induction of flowering. In the rosette dicots, the shoot internodes between the vegetative leaves elongate by the action of the rib meristem activity during the flowering. The activity of the rib meristem derives internode elongation in basipetal developmental gradient, which is highlighted here, with the lower internodes showing maximum elongation as seen in the bar next to the figure. Further, the leaf primordia and floral buds develop from the flanks of the apical meristem. In monocots too, the inflorescence stalks dramatically lengthen due to internode elongation. But this elongation is brought about by the activity of intercalary meristems, which is highlighted here with the IM. Between vegetative nodes, an elongation of the internode occurs in an acropetal direction, which is highlighted here with the arrow. Intercalary proliferation displaces expanding cells upward to regenerate a single, often hollow, internode from the bottom up, which is supported by the enclosing leaf sheath, which is present at the top. Internode lengthening leads to the vertical distribution of leaves, thus exposing the inflorescence. The inflorescence axis bearing floral buds is formed by the activity of apical meristem, and the internodes do not show elongation as the floral buds differentiate. Another interesting growth pattern is observed in the axillary branching, where plants display enormous variation in the shoot body plan, which is both genetically determined and in response to environmental conditions. A major element responsible for the diversity in shoot architecture is the development of axillary meristems, which is highlighted here with the dotted line, which so where the axillary meristems are present between the leaf axis or leaf axils. The axillary meristems have the same potential for shoot growth as the primary sap. Initially, axillary meristems usually generate a small axillary bud consisting of a small shoot axis bearing a few immature leaves that normally we see. The bud can subsequently remain dormant or grow out to produce a branch. So as we very well know that the branching takes place from the axillary positions. The main modes of branching includes the monopodial branching, which is highlighted here, in which the sap produces leaves and axillary branches. And another one is the sympodial branching, where the sap terminates. So nothing but the apical growth is terminated, or we can say decapitated, and the growth of a shoot is carried on usually by an axillary branch at the next lowest node, which replaces the original sap. So such examples we can observe in many plants, like we normally see the ossimum plant. In common, we know that plant is a tulsi. So where if we capitate or decapitate the apical part, the new branches starts coming out from the base. Axillary meristems, which is denoted here with the abbreviation AM, arises from the base of the adaxial side of leaves and give rise to lateral shoot branches. STM expression maintains the meristematic cell fate in the cells of the leaf axils, while Wuschel is required, that is WUS, is required for defining the stem cells. Cytokinin signaling, which is highlighted here, activates the expression of Wush in the leaf axil, which is requisite for AM initiation. A grass family transcription factor, that is lateral suppressor, or LAS, which is highlighted here with the red color, so the abbreviation LAS and RAX is given, which is expressed in the leaf axils and regulates the AM initiation, that is axillary meristem initiation. Loss of function mutants of LAS leads to severe defects in the axillary meristem formation. Other transcription factors called regulator of axillary meristem, that is RAX, are MYB transcription factors, which are expressed in boundary regions of axillary meristems and are specific regulators of axillary meristem initiation, whereas RAX1, 2, 3 triple mutants show an absence of AM development. So both the group of transcription factors are really important for the growth of an axillary meristem. Coming to the second part of the module, which is apical dominance and axillary bud growth, where auxin synthesized by the SAM and young leaves suppresses the outgrowth of a axillary meristem, and this process leads to the expression of apical dominance. So which is highlighted here, where the polar auxin transport mediated by the pin proteins in a downward direction has been known to play an important role in the expression of apical dominance. However, the autofeedback mechanism from main stem and high level of auxin accumulation in the axillary bud, which is highlighted here, induces the bud dormancy. When the auxin level in the main axis are reduced, say by the decapitation, the auxins from the axillary bud flows into the main axis and relieves the bud dormancy. So once the axillary bud auxins are started coming inside the main axis, the dormancy can be broken. Further, cytokinin has been identified as a likely secondary messenger for transmitting the auxin signal to the axillary bud, which is highlighted here. High auxin concentration in the main axis is seen to inhibit the key cytokinin biosynthesis enzyme, that is isopentenyl transferase, that is in short or abbreviation known as a IPT, which is highlighted with the dotted red color line in the nodal region. Due to the inhibition of the cytokinin levels in the main axis, the level of cytokinin in the axillary bud is high, where it brings about the bud activation. So as we can see here, as the cytokinin signaling starts, the bud activation is there in the axillary meristem. Now let's see how axillary bud growth is regulated. While looking at the regulation of an axillary bud or growth of axillary bud, there is a gene known as a BRC1 which plays an important role. So a key branching transcription factor, that is a branched one, in short it is known as a BRC1, is expressed in the axillary buds and is an inhibitor of axillary bud growth. BRC1 mutant, which is highlighted here with the dotted circle, mutants show extensive branching, indicates that the BRC1 negatively regulates the branching. So when there is a presence of a BRC1 gene, the branching will be inhibited, whereas the mutant of a BRC1, which is highlighted here in the figure, shows that the massive branching pattern. Cytokinin is a negative regulator of BRC1. So as we can see here, cytokinin can inhibit the BRC1 and hence cytokinin application promotes the shoot branching, which is commonly observed. The targets of the BRC1 are genes coding for the HDGIP transcription factors called homeobox proteins, which includes the genes like HB21, HB40, and HB53. These HDZIP and BRC1 together upregulates the expression of 9-cis epoxycarotenoid deoxygenase, that is in short known as NCED, which encodes a key enzyme in ABA biosynthesis, that is abscisic acid biosynthesis. High level of ABA promotes the bud dormancy, which is highlighted here. Besides hormones, a transcription factor or a transcription repressor called TIE1, which is highlighted here, is known to regulate shoot branching by repressing BRC1 activity through protein-protein interaction. So as we can see here, TIE1 can inhibit the function of BRC1 and ultimately it can lead to the growth of axillary buds. TIE1 is predominantly expressed in young axillary buds. After formation of the axillary meristem, auxin synthesized in the meristem brings about the breaking of apical dominance by disrupting the basipetal auxin gradient. which leads to the growth of the axillary meristems to form branches. Coming to the vascular development or vascular continuity between the root and shoot, which plays an important role in the survival of a plant, where the plant vascular system performs two key functions. One is delivering resources to plant organs and providing mechanical support. It also acts as a long distance communication network for abiotic and biotic conditions above and below the ground. Vascular continuity is crucial for these functions. In seedlings, the root's primary vascular tissue extend into the shoots, which is highlighted here. The hypocotyle has diarch xylem, which is highlighted in the figure, with the 5 to 6 vessels in a single file. As the seedling grows, parenchymatous elements from the two endarch xylem strands and four phloem strands below the cotyledonary nodes are developed. Higher up, as we move further, the xylem and phloem strands are separated by the pith, forming two distinct xylem strands, each with two phloem strands. These strands extend into the cotyledons as midveins, forming endarch xylem and converging phloem strands to create collateral vascular bundles, which is highlighted here. The vascular tissues are formed by the procambial cells. So here we are going to discuss about the development of phloem, where the vascular tissues are formed by the procambial cells, which besides maintaining their stem cell identity, differentiate into specific vascular cells such as tracheary elements, xylem fiber cells, xylem parenchyma cells, sieve elements, companion cells, phloem parenchyma, and phloem fiber cells. Phloem is made up of sieve elements and companion cells. The phloem precursor cells in the procambium undergo periclinal division to establish the companion cells and sieve elements, which is highlighted here with the S, E, and CC. So S is the sieve elements and CC is the companion cells, which is given in the figure. The phloem early DNA binding with one finger or pair transcription factors mediate early asymmetric divisions in the procambial cells to give rise to the phloem progenitor cells, which is highlighted here. Peer transcription factors bring about the activation of the transcription factor called altered phloem development or APL, which is highlighted with the red color. APL is essential for the differentiation of both Se and Cc, that is sieve elements and companion cells, where it is expressed. APL regulates the sieve elements in enucleation pathway, also inhibits xylem differentiation in the phloem poles. The PS transcription factors also induce clavata3 embryo surrounding region related 25, that is CLE25, which is highlighted here, and CLE26 peptides, which in turn inhibit the expression of PS and phloem formation, forming a negative feedback loop. So as we can see here, CLE25, which inhibits the peer, and whereas the peer activates the CLE25. As the sieve element mature, they experience extensive degradation of their organelles and eventually form a clear transport route. However, the sieve elements still remain living as they retain a plasma membrane and a reduced number of other organelles such as smooth ER, that is endoplasmic reticulum, plastids, and mitochondria. Further, as you can see in the recent figure, where the cell wall of the sieve elements also undergo drastic changes in structure, callose is deposited around the plasmodesmata of a sieve elements, replacing the already present callose. These sieve elements mature, the callose deposits and the middle lamella are removed, thereby forming a sieve plate with enlarged pore at the plasmodesmata sites, which is reflected here. Further, the survival and differentiation of sieve elements depends on a close association with their neighboring companion cells, which are specialized parenchyma cells with an increased number of plastids, mitochondria, and free ribosomes. The companion cells are connected to their adjacent sieve elements by numerous plasmodesmata, which is highlighted in the figure above. Through these connections, the enucleate sieve elements are supplied with energy, assimilates, and macromolecular compounds such as proteins and RNA, which is highlighted in the figure. Coming to the xylem development, where xylem cell differentiation involves temporal and spatial regulation of secondary cell wall deposition, which is highlighted in the figure, where from procambium the xylem cells undergoes in division and it shows the secondary wall deposition and further shows the mature xylem cells. The cortical tubules regulates the spatial pattern of a secondary cell wall by orientating cellulose deposition as discussed. As seen in the module on root development, the class 3 HD zip member functions positively in xylem specification, and the microRNA 165 or 66, which is highlighted in the above figure, is seen to regulate the level of SD-ZIP3 genes. In addition to SD-ZIP3, the vascular related NAC domains or VNDs and the secondary wall associated NAC domain proteins or SNDs function as master transcription factors that can induce xylem cell differentiation. As we can see, these transcription factors plays an important role in the formation of various parts of the xylem or the development of a xylem. As we can see, SD-ZIP3 helps in the metaxylem identity, whereas VND help in the protoxylem identity, whereas SND along with the MYB transcription factors helps in the major secondary wall components or development of a major secondary wall component, namely cellulose, xylan and lignin, whereas VND along with the proteases and nucleases helps in the programmed cell death for the formation of a xylem fiber cells and the tricary elements. In dicot stems, the vascular cambium forms from procambial cells and parenchyma cells in the ground tissue, producing secondary xylem and phloem. As the interfascicular cambium connects and it creates a continuous cambial ring, which is highlighted in the second figure, in radial growth and annual rings. So annual rings which are normally observed in the dicot stem and which is also highlighted in the second figure. The cambium initials divide to form ray and fusiform cells. Ray or fusiform cells which are highlighted in the third figure, and where the ray initials produce radial cells for transport and storage, while fusiform initials create phloem and xylem cells. These cells differentiate into xylem or secondary xylem, wood, and phloem. Sapwood conducts water, and heartwood supports and stores metabolites. Radial growth breaks the epidermis and endodermis, forming the cork cambium from the pericycle. The cork cambium generates protective periderm consisting of suberin and lignin layered dead cells, safeguarding the internal tissue, which is highlighted here. So as we can see here, the secondary phloem and then cambium zone and the xylem is there. So cambium zone adds the secondary phloem and secondary xylem, which is highlighted in the figure. Along with that, in the second figure, which is at the lower position, where which shows the presence of a vascular cambium, above that the cork cambium and the periderm, which is acting as a protective layer for the plants. Coming to the secondary growth in monocots, which is quite different as compared to the dicot plants, where in monocots vascular bundles are scattered throughout the stem, and all cells within monocot vascular bundles differentiate to form closed vascular bundles surrounded by a layer of terminally differentiated sclerenchyma cells, which is highlighted here. And after that, there is no possibility of developing a cambium ring or a cambial ring. As we can see, the vascular bundles are scattered, so it is quite impossible to develop a cambial ring or a continuous cambial ring. In some monocot lineages like asparagales, radial growth of stem is achieved through the action of a lateral meristem termed as monocot cambial, so which is highlighted here in the figure or a third figure which just now displayed. So this is a meristematic layer in the stem that can be recognized from cell files produced by periclinal divisions in the ground tissue. It produces foci of meristematic cells towards the center of the stem, which differentiate into secondary vascular bundles towards the center of the stem, and a parenchymatous secondary cortex towards the outside. The secondary vascular bundles have an amphivessel organization with xylem surrounding the core of a phloem, which is highlighted here. So as we can see here, the amphivessel secondary vascular bundle with xylem, which is surrounding the phloem, which is present in the monocots. During secondary growth in Arabidopsis, coordinated regulation of cambial maintenance is carried out by a signaling mechanism involving a ligand-receptor pair comprising of a ligand called tertiary element differentiation inhibitory factor, that is nothing but a TDIF, and the receptor kinase phloem intercalated with xylem, that is PXY, which is highlighted in the figure. PXY is expressed in vascular tissues throughout the plant body, and its expression localizes to the xylem side of the vascular cambium. Processing of phloem expressed clavata 3 endosperm surrounding the region or CLE protein, which is highlighted here, give rise to the ligand peptide TDIF. Hence, a phloem-derived TDIF signal is important for the activity and bifacial nature of cambial stem cells. Further, TDIF-PXY regulates the expression of Wuschel-related homeobox transcription factor, which is WOX4, in the cambium, which promote vascular cell division and proliferation. This association also includes the expression of zinc finger transcription factor called lateral organ boundaries structural domain, that is LBD4, along the phloem procambium boundary. Through this indirect regulation of LBD4, PXY regulates vascular organization by defining boundaries and amplifying cell divisions on the phloem side of the procambium. As we can see here, the differentiation of a phloem is regulated by the PS transcription factors, which is already seen earlier. PS also induce the CLE peptides, which in turn inhibits the expression of PS, which is highlighted here. So CLE plays a negative feedback loop. And in addition, PXY along with the glycogen synthase kinase 3, that is GSK3 family proteins, negatively regulates the differentiation of a vascular stem cell into xylem. So it can inhibit the vascular xylem differentiation. The binding of TDIF to PXY leads to the dissociation of the PXY GSK3 complex, which results in the promotion of a xylem differentiation. Further, the VND6 or VND7 transcription factors and the SND1 transcription factors regulate the differentiation of a metaxylem and protoxylem and the formation of a secondary walls as seen earlier in this module. To summarize, we have seen in this module how the shoot grows through the activity of the shoot apical meristem and axillary meristems to form the distinct architecture of plants. The shoot shows a vascular system that connects the root to the leaf and other above ground organs and plays a role in conducting water, nutrients, photosynthesis, and signaling molecules throughout the plant. The vascular system also gives support to the growing shoot by bringing about an increase in the stem diameter. In the next module, we will see how leaves develop on the shoot and the leaf development will be discussed in detail. Thank you.